So I'd like to briefly describe to you what we did in class today. So students got this sheet and we talked about what a karyotype was, and we've mentioned this prior, but we were kind of digging in a little bit that if you get a sample of cells and you find a cell that was just about to finish cell division, so one cell completing the mitotic process, so going through mitosis, the cell cycle of mitosis, and all those chromosomes had doubled up and then conjoined. Your chromosomes will look like these two sister chromatids joined right at the middle. And you can actually organize all of these chromosomes via computer. We used to do it with a photograph and scissors, but now we use the computer. We could actually arrange all of one human's chromosomes together. We arranged them in order of size and of banding pattern, how it was stained, and what these things actually code for. So you'll see the first pair of chromosomes is the biggest, and as you go down all the way to the 22nd pair, they're the smallest. And then you end up with the last pair, the 23rd pair of chromosomes for people. And these can determine, in part, your genetic sex, genetic male structures or genetic female structures. Again, this sex is so much more detailed than just what these chromosomes are coding for. There's multiple other locations that can help influence what our sexuality is, but this XX and XY is the traditional genetic pattern in your chromosomes for male and female. So down at the bottom, we're looking at those genetic typical patterns. So we talked about this a little bit, and why someone would try and take a look at their chromosomes would give them a pretty good picture of what might be happening in their lives. Or if it was a developing fetus, we could take a look at that and see if this little fetus needs a little extra support when the fetus is actually done developing and is born. Gives the family members a little bit more opportunity to prepare for an atypical birth. So where we're starting is at the top of the next page. This is typically what a photograph of the chromosomes would look like inside of the nucleus. And so digitally through a computer or old school by hand and scissors, which is one of the things we're going to do in class, you cut out all these chromosomes and then you arrange them simply by the banding pattern and by size. And so what we used was a blue or green sheet that had a whole bunch of background information on it. And so together we went through all of these practice karyotypes. So the first thing you want to do is take a look and see if you can find that last pair of chromosomes and then determine if it's male or female. So this is two chromosomes where there's supposed to be a pair. So there's 1, 2. One of them is a really tall one. You can see real close. And then the other one is a really, really short one. The first one is an X. X are quite large. And the second one is a Y. So we would say this is an XY, which makes them a genetic male. And then you look to see if all the patterns, if all the pairs of chromosomes seem to be present and structurally similar. So as you're looking, there's a little bit of slight variation, but nothing dramatic. So we would call this a normal male presentation. Come down and see the next one. The next one we were looking again for our 23rd pair, and we actually have 3 chromosomes for the sex chromosomes. So that means we have an X, and a Y, which means we've got a genetic atypical presentation. This is not standard. So you can see XXY, that means you've got 3 chromosomes where there's supposed to only be 2. So we'll say 47 instead of the normal 46, and we'll say there's an extra sex chromosome. We'll learn more about this when we get into the specifics of our reference sheet, our blue or green sheet. Let's take a look at the next one. Again, we're going to go find our sex chromosomes, that last pair. This has just got 2 X's, no Y's, so that means it's female. And then if you look at all the pairs, even the ones that look like they're dancing, they look really, really similar. Little slight variations, but for the most part, they're all matched up with an exception here. Here we have 3 chromosomes. That's the 21st pair of chromosomes, has a triplet. We'll actually call this, when there's one extra at this position, a trisomy. And we'll say which pair it's occurring at, trisomy 21, and then we'll say if it's male or female. And this would be a female. I'd like you to try the rest of these. You can see the answers here as we move through it. And then in class, after we did this practice together, students got 6 more karyotypes and then a big blue reference sheet to try out. So good luck, and send an email if you have any questions at all.